So today we are meeting virtually to have a candid discussion at the 5th Pharma Pricing, Reimbursement, and Market Access 2021 conference, as we continue our attempts to make the virtual conference platform work in terms of networking with new people and discussing novel ideas and solutions to various issues that we are attempting to tackle in our professional worlds. There's this lingering memory that most of us have of casually meeting another person or a group of people at a conference, attempting to read name tags, and at times totally failing miserably, and just having candid but brilliant discussions about the most relevant topics of the time. Obviously, the global pandemic has stopped those organic discussions from taking place, but in the memory of candid in-person professional discussions, I would like to thank all of you for taking the time out of your busy schedules to join me on this virtual platform today. Before we get started, why don't we do some quick introductions?
Hi, I'm Katie Clapp. I'm president and co-founder of FRAXA Research Foundation, which is a nonprofit that my husband, Mike Tranfaglia, and I founded back in 1994 when our son, Andy, was diagnosed with Fragile X syndrome. So I'm thrilled to be here today.
Hi, I'm Mike Tranthalia, and I'm the Medical Director and Chief Scientific Officer at FRAXA Research Foundation. As Katie said, one of the co-founders of FRAXA, and I'm also a psychiatrist, and I treat patients with Fragile X, and I'm a Fragile X parent. My FRAXA duties include supervising the research portfolio of FRAXA, which has become increasingly preclinical and clinical in its orientation, with a lot of clinical trials of investigational new drugs and repurposed existing drugs in Fragile X.
I'm Christopher Missling. I'm president and CEO of Anavex Life Sciences. We focus on developmental disorders, but also neurodegenerative disorders. And I'm very excited today to talk about Fragile X, specifically today with the FRAXA Foundation.
My name is Nadia Bodkin, and I do wear several hats throughout the rare disease ecosystem, but for the purpose of this conference, I am the co-founder of the Rare Advocacy Movement, RAM, which is the first community-based rare disease activist network of professionals organized to benefit the interests of people with lived rare disease experiences. By way of education, I have my doctorates in pharmacy and master's in genetics, but that's quite honestly not the reason why I've become so heavily involved in the rare disease drug development industry. The true reason behind my rare disease passion is that I personally was born with 3 rare genetic conditions: a connective tissue disorder called Ehlers-Danlos syndrome, a blood disorder called beta thalassemia, and as if that wasn't enough, I also have alpha-1 antitrypsin deficiency. And the combination of the 3 have led to several comorbidities, as I'm sure all of you can imagine. So in the interest of time, today we would like to discuss the novel framework in which an advocacy or community-based organization is able to effectively collaborate with a biopharmaceutical drug developer and vice versa at the preclinical stage of drug development. And there's this old, extremely outdated preconceived notion within the drug development industry that opportunities for patient-centric collaborations don't really exist until the commercial phase of drug development. And while several patient advocates and community activists have been demanding more effort be made by industry stakeholders to incorporate the patient perspective into clinical trial design and protocols, both of your organizations went a step even further and established a preclinical collaboration, which is extremely novel in today's environment. Katie and Mike, can you give us a brief description of Fragile X syndrome and how it impacts the people affected?
I'll take it from the formal medical end, and then maybe Katie can add some personal details to that. But Fragile X syndrome is, first of all, the most common inherited cause of intellectual disability and autism. It's a major neurodevelopmental disorder, and it's a developmental disorder in the true sense of the term, and that people with Fragile X don't really have very much wrong with them medically, and they tend to live a normal lifespan, but they just don't develop at a normal rate. So Fragile X directly impairs the mechanisms that are responsible for development of the brain. Fragile X is, as I mentioned, one of the most important models for studying autism, and about 30% of the males with Fragile X meet full criteria for autistic disorder, and just about everybody with the full mutation of Fragile X has something that we would now term on the spectrum, autism spectrum disorder. So it's a really important monogenic condition despite the name, which is a little bit of a misnomer, it's not a chromosomal disorder, it's a single gene. And it was actually the first of the trinucleotide repeat disorders that was discovered. So Fragile X is caused by an expansion in the promoter region of the FMR1 gene. And there's a CGG repeats, which in normal folks are usually around 30 repeats, but become wildly expanded to over 200, sometimes over 2,000 repeats in Fragile X. And that has the net effect of silencing the gene. And curiously, because the defect, the mutation, is in the promoter region of the gene, the coding portion of the FMR1 gene is entirely normal. And so there's the prospect out there someday, somehow, of reactivating the gene, because everyone who has Fragile X actually has the proper genetic code. They're just not using it.
So I'll add that our son, Andy, has Fragile X syndrome. And that's, of course, the reason why we started FRAXA to accelerate research. Now, Andy is 32 years old now. He lives at home. He's quite impaired in a way he's very typical of males with Fragile X, who tend to be more impaired than females. He's able-bodied, a little klutzy, little bit of anxiety walking around, you know, trying to go down hills, little sense of inability to navigate, you know, couldn't climb a mountain for sure, or if he could, he would never get down. He can speak, but it's just, you know, small sentences. Has quite a lot of OCD, anxieties. He's got quite a few challenges, including occasional seizures, which also can be related to Fragile X. So that said, he also has really good days where you can see the potential when he's thinking clearly, communicating well, really happy, cooperative. And so there's most definitely potential for him to perform a lot better than he does if we got the right treatments on board.
So collaborative partnerships like this one always have more than one perspective as to its origins. So how did your organization come to the point of being able to connect and launch this novel approach to community-based drug development?
We've had a lot of success over the years sponsoring academic research, and we've had a lot of success on the scientific front in understanding some of the basic mechanisms of disease in Fragile X. And along the way, as we've come to understand the basic mechanisms, we've identified potential treatment targets. But we found ourselves running into a roadblock when it came to doing preclinical validation, when it came to actually testing drugs or testing novel compounds in Fragile X animal models. It's often really difficult to get academic labs to do that. You can imagine that if you have a lab that's set up to do molecular biology, they're not used to doing live animal work, for example. They don't know how to handle mice. They might not have the facilities for it. They certainly don't have the expertise in doing it. So we encountered this roadblock at the preclinical validation stage. And a long time ago, we came up with one solution, which was to sponsor a lab that would do contract research for us. And we could say to the investigator at that lab, here's a compound that we find interesting, and we think this might be therapeutic for Fragile X. Nobody else in the field is working on this right now, but could you test it in the animal models and see if you can get some useful responses from this compound? And as we did more and more of that, we started to recognize the value in doing that. It's incredibly important in de-risking whatever particular therapeutic approach you might be pursuing, and in drawing in industry partners. And so over the years, we've beefed up that capacity, and we now refer to this as the Drug Validation Initiative at FRAXA. And we're actually on version 2.0. The the original investigator retired, but we've got a new facility, and it's worked out tremendously well. And it's something that we can offer to pharma partners, because a lot of the partners that we work with, they're very small companies. They certainly don't have in-house biology labs. They can't always afford to contract with a traditional CRO to do this sort of work, and it can get pretty expensive. But we have this facility up and running, and it has the additional benefit of doing the validation the same way every time. We have the experience of testing multiple compounds, some of them available drugs, some of them investigational drugs, but we've used the same protocols and the same mouse colony for all of them, so we can actually do comparisons between different types of treatment approaches to see what works best. And importantly, now we can also do some really complicated things like investigating combinations of medications, which would be really difficult to do in any kind of academic lab.
Wow, that's really amazing. The drug validation initiative, that sounds like something that a blueprint that the general rare disease space could totally utilize.
I think it's one of the most important things that we do, and I think it's something that every rare disease foundation should do. It takes a lot of hunting around, and it's only because we've funded lots and lots of different academic labs that we found the right situation. It's not easy to come up with the right facility. For example, most American universities, they have rather difficult tech transfer departments that want you to sign all kinds of contracts that require a lot of negotiation. So anytime you want to test somebody else's proprietary compound in an academic lab, there's a period of negotiations. And we've had cases where we were trying to negotiate one of these materials transfer agreements with an academic lab that we were working with. And one of our pharma partners. And it went on for 18 months and ended up with no resolution. We never actually did the research there because they just couldn't come to terms, because basically the universities see this as a profit center, that they'll get a stake in the intellectual property. But of course the companies think, this is our property, you can't have any part of it. And they don't want to sign those contracts. And so you need to come up with another arrangement. And what we've got now is a lab that's outside of the US, in a country that is a lot easier to deal with. The universities don't have the same ideas about intellectual property and potential revenue streams emerging from the research that they do. So it's worked out really well from that point of view.
One reason that this all came up, or we didn't start out with a FRAXA DVI for these studies. We would be back at the real meetings when you actually got to sit with people for 3 days and talk. And there would be so many investigators that we'd funded to do a mouse study, and they were having terrible problems with the Fragile X mice who would not breathe. They would not, behave. It turns out that if they're by the door in the lab and there's a breeze or people are opening the door, they get anxious, and so they don't do what they would do otherwise. So there's all this expertise that had to be shared by the experienced Fragile X mice people who knew them. So this is another way around: quicker, easier, cheaper, and more valid, because the studies turn out to, they're done the same way and they can be compared.
Wow.
It's
So
been a wonderful study.
what you've done is incredible. And so now this drug validation initiative, we have some insight on how it came to be. And we have Dr. Missling here. Can you please provide us with some sort of insight on how it's been on the other side, on the biopharmaceutical side, to participate in this program?
Yeah, absolutely. So we had developed our first drug, in the pipeline, which was called Anavex 273. And we had just initiated the first clinical study in a neurodegenerative disease, which is Alzheimer's disease. So while this was on autopilot almost, I was looking for what else could this drug do in terms of helping patients, since the mode of action was intrinsic to chromatin remodeling, which is something which Mike just explained, which applies to Fragile X syndrome. And so we really were looking for websites and found the FRAXA Foundation website. And literally, we clicked on it and it said, if you have a compound, you can, send it to us and you get it tested. And we at a third-party lab, and we didn't have a laboratory anymore. And it's the best way you can find, because I trust Fragile X Foundation. FRAXA has the best model developed because they're really intrinsically interested in developing a drug for exactly that unmet need for that disease. And so we had the fact that there's somebody does that who is the most expert with the most expertise, but also by a laboratory which is completely independent without any, subjective interest about the outcome. And so we really, send the compound over, and lo and behold, I think we got a call from Mike. He said, "What you got here?" And we said, "Well, it's just our compound. We thought it could be doing something in Alzheimer's. Now we want to see another indication." That, well, it's really did something, And from there, we were really intrigued that the data was so phenomenal that we just were able to publish this data actually last month, and in a very high visible, peer-reviewed paper. And it really shows that the drug did reverse the symptoms of the Fragile X in animal models. And that led us now to move ahead with planning a clinical study in Fragile X. And I like to also mention to inquire and talk to Mike again, to also be connected with families and Katie, and see how we can now design together the clinical study to make it the most easy for patients and for the families to be part of such a study. So this is the next level of interaction I'm planning to have with Mike and Katie in order to move this forward into the clinic. And that's right now the plan.
You pretty much summed it up. This is a brilliant example of preclinical collaborations, community-based collaborations that are now setting the scene for the clinical setting and actual clinical participation from the community in designing those protocols. Katie and Mike, how's your organization managing the transition from now you have a successful preclinical program through the drug validation initiative, your transition to now to contribute to the clinical protocols? Is there another program that you transition into?
This is, in our minds, just part of the overall task that we took on when we started FRAXA, which is to go from where we are to treatments for our kids and others like them. So, back when we started it in the 90s, there was no mouse model being shared with the community. Then there were a lack of antibodies. The bottleneck traveled along the drug development pipeline. And we are actually now really at the final step on that pathway for many potential treatments, and that's the clinical trial. So FRAXA does an enormous amount of clinical work. We fund clinical trials. We don't have a lot of money, so these tend to be pilot trials. We just finished funding a phase 2 trial that was very successful. And so now we're working with the partners, the pharma partners, on the phase 3 trial plans. So this is definitely just part of the system. And I guess we could call it another program, but FRAXA is small. We're small, but we're focused. And we absolutely have many ideas about how to design those trials. We think about it every day. How does Andy, our son, take pills? Could we get him to the hospital for blood draws? What about placebo effects? All of those things, because we've been doing this for many years, for many trials. All of our kids have been in trials and will be in more. So there's quite a community. Part of what we do is let the community know, we can recruit hundreds of Fragile X families. They are so eager, and they've been learning along with us all this time. So they're eager, they trust, and they're good partners.
Clinical trials for rare diseases are a real challenge for a couple of reasons. First of all, it's a rare condition, so you don't necessarily have a lot of potential volunteers for your clinical trial. I guess we're lucky in the sense that we're just about the most common rare disease around. So we actually have plenty of trial subjects, and we don't have too much trouble recruiting for the size trials that we need to do. But for example, I'm on the scientific advisory board of a foundation for another rare disease that has about 500 people total in the United States. And if you think about that, it's an average of 10 people per state. How do you get a clinical trial going, especially because they're all different ages and everything? So it's really difficult. And groups like that face a different kind of challenge. But in our case, we have enough trial subjects. It's getting the experience in actually doing the clinical trials. And another problem that rare disease foundations typically have is that there is not a well-worn path in drug development for that disorder. You need to come up with endpoints that are going to be useful for your trial. You need to have trial sites that are going to be able to conduct the trial in a rigorous way. And it's like anything else. How do you get good at doing anything? You practice, you do it over and over and over again. And so that's been our approach for the most part to clinical trials, is to get better at doing clinical trials by doing clinical trials and by doing more clinical trials. And at this point, we've done quite a few. And some folks may know that there were large international clinical trials of mGlu5 antagonists by some major pharma companies a few years back. And that helped to develop a lot of the infrastructure. So we had devoted a lot of time and effort to training up some excellent clinical researchers. In fact, they're some of the top clinical researchers in the Fragile X field now. And they got started with FRAXA grants because we knew we needed to bring people into the field. We didn't quite realize how long it was going to take, but since we've been at it for a while now, we've actually got a large number of clinical investigators that are trained and ready to go when a promising new treatment strategy comes along. And so that's an important concept. You need to actually think about doing clinical trials. If you're a rare disease foundation, and I talk to people every week that are just starting a rare disease foundation, you need to start doing clinical trials right now. Obviously, you can't do a clinical trial of an investigational drug that's based on an elaborate understanding of the mechanism of action or the mechanism of disease, but you can start with repurposed available drugs in your clinical trial population, and you can start to develop that capacity. And that's a really important thing to do for the rare disease foundations.
So the overall goal of preclinical collaborations like this one is to encourage and speed the development of new, safe, and effective therapies for rare conditions like Fragile X syndrome with high unmet medical needs. And as a member of the rare advocacy movement and as a community-based activist myself, I would just like to applaud the FRAXA Foundation for developing the drug validation initiative and for Anavex for participating. And now that all the pharmacokinetics and preclinical data supports movement into clinical trials, it is really encouraging to know that your organization has chosen to develop this novel framework of collaborative community-based drug development. And as this collaboration continues to evolve, I look forward to future discussions that unveil the lessons learned from this novel approach. So before we close our discussion, it would be really great if all three of you could leave us with one message or lesson or goal that you would like for the rare disease drug development ecosystem to receive and take into consideration.
Well, as a mother of somebody who has Fragile X syndrome and who is challenged by it, it is incredibly exciting to me to see what we can accomplish by working together. When I first got into this, I had no idea just how dynamic the pharma field is and pharma scientists are, and how motivated virtually all of them are. So it's been a pleasure. It's been exciting, and I have no doubt that we will be celebrating a success.
I just want to encourage anybody out there in the rare disease world to swing for the seats, to really try to do it. We see a lot of foundations that want to increase awareness or provide support, but they think it's too much to actually try to cure this disease of theirs. And I just want to say that it's not too hard. You can do it. We don't have any special resources. We don't have billionaires backing us up. We just have the community sending in small donations here and there. And we don't have a huge research budget either. We've got more than we used to have. We've built it up over time, but it's hard to raise money and it's expensive to do research. But the stuff that we've done has made a real difference. And I would encourage any groups out there that are maybe on the fence to just do it and realize that you're not going to necessarily succeed the first time around. It takes a couple of tries to get it right. You have to learn what you're doing. But if you don't get out there and do it and make the mistakes, you'll never get there. So my motto going forward now is realizing that we need more shots on goal. We need more attempts at treatments for Fragile X. We need to spin this development cycle faster and faster. And part of that is that you have to try and you have to fail. And what we're trying to do now is just fail faster and try more times. So get out there and do it.
I'm very grateful, as our team is, to Mike's and Katie's enthusiasm to give us a chance to try. And without FRAXA, we wouldn't be here talking and providing this data we have. And what I also would add to that, what we found in this collaboration was not only we found that the data was very encouraging, but also we found a biomarker of response, which now becomes more and more important, especially in CNS indications, where there is more fluctuation in the outcomes and variability inherent. And that makes it even more important to connect or lock in the outcome with a biomarker of response. And given of the work we did with FRAXA, we were able to confirm and identify and confirm that. So which will give us additional support in the ongoing and planned clinical study for Fragile X. So again, my thanks to FRAXA Foundation.
You guys are incredible. I'm so honored to just be on this virtual stage with all of you.
Well, it's been a pleasure, really.
A pleasure.
Exciting.
Likewise.
This is very exciting.
Thanks for inviting us.
Bye.
Bye.
Bye.